Hi dear students, welcome back to one more session of immunology. Today I would like to introduce you to a term called the antigen. Quite often when we consider the subject of immunology, we use the term antigen. But what is an antigen? You need to know that. Any self as well as a non-self substance which can elicit or induce an immune response and react with the products of that response is called an antigen. Now, what do you mean by an immune response? An immune response is something in which what happens? Either the B cell is interacting with an antigen or a T cell is interacting with an antigen, and it is a type of protective mechanism which will is being produced within your body when something which is not supposed to be there in your body has come. So, antigen, it refers towards to any substance which will cause your B cells or your T cells to get activated and either produce antibodies or either act as effector cells. So, sometimes we can see that self molecules and non self substances. Now, non self is quite clear to us. Substances which are not within our body, if it comes from outside, they can induce an immune response. But sometimes, in certain cases, even our bodily self surface antigens, due to some changes or due to some errors, they can induce an immune response within our body. So, the term antigen comprehensively includes all those self as well as non-self substances which can induce an immune response within us and also which could interact or react with the products of that particular response. That is referred to as an antigen. Now, what is the nature of this antigen? It could be either a protein, a nuclear protein, a polysaccharide, or sometimes even glycolipids. So, the term immunogen or the immunity generator is a more precise description for a substance which could elicit a specific immune response. And though we use the term antigen more frequently, the term immunogen is a one which is better to include them under the term rather than having antigens. But most frequently we use the term antigens. So if any substance, if it induces an immune response and react with the products of that response, then we call them as a antigen. Now, as I told you, either it can be a B cell mediated immune response or it could either be a T cell mediated immune response also. Now we will just see what are the substances which could induce a immune response within us. As we all know, whole cells and viruses are good immunogens. Now you can have bacteria or you can have viruses, or sometimes even if we put unmatched blood within our body also, it will trigger a immune response within our body. So whole cells either of foreign particles of bacteria, viruses, and even unmatched blood groups, they could serve as good immunogens. And in some cases, you can see that complex molecules with several epitopes could also serve as immunogens. Now, what is an epitope? We will talk about it. See, if you consider a particular molecule, okay, if you consider the surface molecule or the part of the foreign particle which is interacting with the antibody is referred to as the epitope. So if you have a complex molecule with having different epitopes, now if you consider this particular molecule, see this blue color is one type of epitope. Okay, this triangular shaped is one type of is an epitope. Then that is, if you consider the flagella itself is having lots of epitopes. Or if you go to consider this surface, that itself is having different types of surface molecules onto it. So, a complex molecule with different surface molecules on it or different epitopes on it will serve as a good immunogen. That is, a complex molecule with different epitopes could trigger a better immune response within you. Okay. And if you go to consider poor immunogens, they could include molecules which are either which are simple molecules. They could be either simple molecules, or they could be even large repetitive molecules, which could large and repetitive molecules could also sometimes cause poor immunogens. So either when they are being attached to a carrier molecule, or when they are simple or large repetitive molecules, they are being considered to be as what? They are considered to be as poor immunogens. Now, if you go to consider the term epitope and paratop more precisely, the epitopes are the regions or the sites of the antigen that will bind to a specific antibody or a T cell receptor. Chemically, epitopes could either include sugars, organic acids, or bases, amino acid side chains, hydrocarbons, and aromatic groups. So I'll just give you an example. If you take a particular virus. Now, for example, we have COVID-19 here. In the case of COVID-19, on the top of it, you have certain spikes attached to it. And the spike protein can be interacting with the antibodies. So, if the spike protein is interacting with an antibody, the spike protein is considered to be as a epitope. And so that is a epitope, the part of the antigen which will interact with a specific antibody or the T cell receptor is referred to as an epitope. And the part of the antibody which interacts with the antigen is called the paratop. So if you have this as an antigen, only some portion will interact with the antibody. This is an antibody. And the portion of the antigen which interacts with the antibody is referred to as the epitope. And the portion of the antibody which interacts with the antigen is referred to as a paratop. And if you go to consider chemically, it could be either proteins, it could be sugars, organic acids or bases, hydrocarbons or even aromatic groups. And in addition to this, you can also see that there would be hidden epitopes also. Now, only the surface molecules or the outside type molecules which will interact with the antibodies are considered here. But there would be other functional, other than these are called the functional epitopes. There would be also certain hidden epitopes also. So, and the number of epitopes is being considered to be as the valency. Okay, so the functional valency of BSA, it has a valency of 6, but the enzymatic digestion shows that it has a total valency of 18. So in the case of BSA is a protein, okay, and BSA can interact with in fact 6 antibody molecules, and at 6 sites it can interact. So the valency of BSA is considered to be as 6. But if you digest it, you can see that it is having a total valency of about 18 also. So the number of regions or the number of sites at which the antigen interacts with a specific antibody will refer towards its valency. Now, if you go to consider the T cells and the B cells, they interact with the antigens in different manners. And there is a antigen recognition by the T cells and the B cells is being found to be different when you consider the B cells and the T cells. As we all know, the B cells, it mainly involves the interaction of the membrane-bound immunoglobulin and the antigen. And in the T cells, as I told you, you always require the presence of a MHC molecule to present the antigen towards the T cells. In the case of B cells, soluble antigens can directly interact with the antibodies. And but soluble antigens cannot bind with the T lymphocytes. And the involvement of MHC molecules is not required in the case of B cells, but it is required in the case of, that is, MHC interaction is needed in the case of T lymphocytes. And the chemical nature of the antigen could be protein, polysaccharide or lipid when we consider the B cells. And if you consider the antigens which interact with the T cells, they would be mostly considered to be as proteins, but sometimes lipids and glycolipids presented on the MHC-like molecules can also interact with the T cells. And if you go to consider the epitope properties, do remember what is an epitope. If you consider a whole antigen, only a small portion of the antigen will interact with the antibody, or the sites which interact with the antibody, they are referred to as the epitopes. In the case of the antigens recognized by the B cells, they are accessible, hydrophilic, and mobile peptide containing sequential or non-sequential amino acids. They are mobile peptides which will contain sequential or non-sequential amino acids. But T cells can interact with internal linear peptides produced by the processing of antigen and bound to the MHC molecules. So that is what is an antigen. You got what is an antigen or an immunogen, and in the immunogen, we know that not all portions of the antigen or the immunogen interacts, only the epitope portion interacts, and based upon this, we call the number of epitopes which can interact with the antibody is referred to as the valency, and we know there would be sometimes a hidden valency also for some people, for some antigens, and the type of antigen interaction with the B cells as well as the T cells will be found to be different. Depending upon the type of antigen present, either the B cell or the T cell will be found to be interacting with it. Now, so let me just talk about the valency once more. The number of epitopes on the surface of the antigen is called its valency, and the valency, it determines the number of antibody molecules with which the antigen can combine at one particular time. Now, if you go to consider an antigen can interact with only one antibody, then it is called as a monovalent, and some antigens can interact with many number of, okay, they will have more than one copy of the same epitope, and hence they are considered to be as multivalent. And multivalent antigens will generate or elicit a stronger immune response that are considered to be as than the monovalent antigens. And the functional valency is different from the total valency of an antigen. And as I told you, bovine serum albumin or BSA is having a functional valency of 6, but the total valency of 18 is being found when you digest it with a particular enzyme. Now, next I would like to talk about what are the factors which will influence this immunogenicity or antigenicity of a particular substance. If you go to consider an antigen, its immunogenicity depends upon various factors. That is, the ability with which it can trigger an immune response, it depends upon various factors. The first one is its size. The most active immunogens tend to have a molecular weight of a mass of about 100,000 Daltons. Okay, and if you go to consider the ones which are below that, what happens? They will be what? Less than 1,000 Daltons. If you go to consider, they will be poor immunogens. And the substances between 5,000 to 10,000 Daltons are poor immunogens. So if a antigen is having a high molecular mass of about 100,000 Daltons, then it is a good immunogen. And if it is less than 1,000 Dalton, it is a poor immunogen. It is not a very good, it's not, it has been found to be what? Not much immunogenic. Okay. And so size plays an important role. You have tetanus as well as insulin toxins, which are examples to prove this factor. Now, the second thing which we consider is the chemical nature, structural complexity, as well as the solubility of the particle. If you go to consider the different particles, you see that proteins are being found to be more immunogenic than polysaccharides, and polysaccharides, okay, is being found to be what? It is more immunogenic than the lipids. So, protein is being found to be highly immunogenic than the polysaccharides, and polysaccharides is immunogenic than that of the lipids. So the best one, if a foreign particle or if an antigen is being found to be highly proteinaceous in nature, its immunogenicity is being found to be higher. And you can see that the presence of aromatic amino acids like tyrosine will increase the increase the immunogenic or the immunogenicity or the antigenicity. So in the protein itself, if you have tyrosine and various other aromatic amino acids, its ability to trigger the immune response is being found to be higher. And the chemical complexity will increase immunogenicity, and sometimes the synthetic homopolymers will tend to lack immunogenicity regardless of their size. So if they are homopolymers, if they are being found to be synthetic, sometimes they would lack in immunogenicity. And the studies have also shown that copolymers consist of different amino acids or sugars are being found to be more immunogenic than homopolymers. So as the structural diversity or the structural complexity of a substance is being found to be increased, it will cause more immunogenicity. And the polymers not soluble in the body fluids are not antigenic, and any substance that is being converted into a soluble form by the tissue enzyme is also not being found to be antigenic. So if you put into your body fluid any substance which is not soluble into it, it will not become antigenic. Now, for example, I can tell you something. If you put a steel in your body, okay, it's not a body fluid, okay, but within your tissue, as a bone support or something, we might sometimes put a iron bar or a iron the thing to support our bones, but that is not antigenic because it is not soluble in a body fluid, and it is not also being digested by our enzymes. So in such cases, substances which are not soluble in a body fluid and which are not degraded by our tissue enzymes are not immunogenic, or they will not trigger an immune response within us. Then the third factor is the foreignness of the substance. If you go to consider, the more foreign it is, the degree of immunogenicity has been found to be increased. Now, if two substances are being found to be highly distant between, that is, if the phylogenetic relationship is being found to be very much, there is a large phylogenetic distance between two species, then for example, then the antigenic disparity between them is being found to be different. So it has been found to be greater. That is, the more farther it is, they don't have much similarity, or the foreignness has been increased. So the immunogenicity is being found to be what? Greater. So the antigens that are not related to us, if that they come, okay, then we have a better immunogenicity, or it will trigger an immune response in us. And coming to the next factor, if you consider bovine serum albumin, it is not immunogenic when injected into a cow, but it is strongly immunogenic when it is injected into a rabbit. This is an example. Bovine is coming from a cow. So when it is injected into a cow, it is not immunogenic, but into a rabbit, it is injected, it triggers an immune response. And some macromolecules, like if you consider collagen and cytochrome C, they have highly conserved throughout evolution, and you can see that they display very little immunogenicity across diverse species lines. So, like if you some molecules are being found throughout in different diverse species. So, collagen and cytochrome C and all has been present in almost all of the species. So, they are of least immunogenicity across the different species. But you have also certain other components. Now, for example, our cornea as well as the sperm, which is being found in the humans, they are not being exposed to the immune system throughout the lifespan. So what happens is, if these tissues are injected into an animal from which they are being originated, and then they will serve as immunogens. That is, coming to the cornea tissue as well as the sperm, they are totally kept away from our immune system, or the immunological cells do not come into contact with these corneal tissue and the sperm cell during our lifespan. So though they are of our body, if you take them and you put them into your blood or into any other place where the immune system has been active, they would react. So in such cases, they will function as a immunogen also. Then the immunogenicity, it also plays an important role at the root of experimental injection. The dose as well as, and the dose also would play an important role in immunogenicity. If you put an insufficient dose for an immune response. Sometimes it will not trigger an immune response. Only if a sufficient amount of antigen is being put correctly, probably only then you would get a, like if you are trying to inject an antigen into an animal and trigger an immune response, only if a sufficient dose has been given, the immune response has been triggered. And the route of injection also plays an important role. So you can either inject them through the vein, then or through the skin, or beneath the skin, or through the muscle, or intraperitoneally. So depending upon this route also, the immunogenicity will be found to be varied. Okay. You can see that the antigen administered intravenously is directly carried to the spleen. It is carried to the spleen. And if you go to consider, if you inject it subcutaneously, it goes to the lymph nodes. And differences in these lymphoid cells and that populate these organs will reflect in the immune response. So, and in some cases, we would also like when you're trying to, like when you want to generate a vaccine and all like that, you would give a what you would want to trigger the immune response. So for that, what happens, you will take the antigen and you will mix it with some particles called the adjuvants, and so that the immune response is increased. uh, Adjuvant is a substance which, when mixed with an antigen and injected with it, enhances the immunogenicity of that particular antigen. Now, for example, we have alum, Fruins adjuvant, etc. They are often used to boost the immune response when an antigen with low immunogenicity is being used, or when only small amounts of an antigens are available. We also give adjuvants along with it so that the immunogenicity of that particular substance is increased. And these are some examples in which you can see that when these adjuvants are being given, the immunogenicity is also increased. So I guess you got an idea about what is an antigen and what are the factors which affects its immunogenicity, and how can we improve the immunogenicity of some antigens by the usage of adjuvants. With this, we come to an end of the first session of antigens. Thank you for now.